All right. So sequencing genomes is getting more and more important, and the technology is getting faster and cheaper. Every couple of years, there is significant improvement. So like I say, I would not be surprised that within 10 years, maybe 15, we will have the ability to sequence entire genomes right here at this little small regional university. It will be a kind of price that a place like this can afford. So that'd be pretty cool. That'll open a whole new avenues for our students. Okay, well anyway, so that's some important types of things. Okay, now a couple other things I want to mention. A modification of this hybridization technology has gotten very, very important lately. It's what we call, now we can't do this here, we don't have this. It's called microarrays. And it's a very powerful modification of nucleic acid hybridization technology. What we do is this. Take cells or tissues or what have you. Extract the total messenger RNAs from those cells or tissues. We use reverse transcriptase to make our complementary DNAs of every single messenger RNA that happens to be found in that cell type. Okay, meanwhile, what we have is this big, huge plate with little tiny wells in it. Hundreds, even thousands. We make sure our cDNAs are labeled. Okay, fluorescent dyes are typical nowadays. Okay, now, this is what we call the microarray. Well, what's in each one of these wells? Each one of these wells has sticking tightly to some kind of matrix of the well so it doesn't float off. It's got a little stretch of DNA from a particular gene. In other words, effectively, it's got a probe to every single gene, or at least every single, to each well having a probe to a different gene. And it's attached to the surface so it doesn't fall off. Okay, so we got a probe here, gene A, gene B, gene C, gene D, gene E, and so on down the way. Each well has a probe to a specific gene. Now what we have are labeled copies of every single messenger RNA that cell type produced. We added here standard hybridization technology. If those cDNAs, the labeled cDNAs, they're copies of all the messenger RNAs that were in that cell type, if they meet its complementary DNA probe here, it will stick to it. And that well will glow in the dark or give you a stain or wherever the case is. Yeah?
Did you say that the whales, are they gel or milk?
No, it's actually usually a little plastic plate that's about this big, and with a lot of tiny little wells, and anywhere from several score to several hundred each, and then you may have a whole bunch of different plates, and overall you may have, for instance, several thousand genes, probes to several thousand different genes in this collection of these plates here. So they're plastic things, generally about this big. Okay, well, all right. Now, anytime one of these cDNAs is complementary to any of those probes in this microarray, it's going to stick and it'll glow at you. So, when you're finished with this, you find this lights up, this lights up, this lights up, this lights up a little bit, but not much, this lights up, and these others don't. What this allows you to do, these microarrays allow you to do, What it allows you to do is this. It allows you to detect every single expressed gene in that sample. So you take, say, a cell, let's suppose a neurite, a precursor of a nerve cell, and you say, what messenger RNAs are produced in that, and compare that to a mature nerve cell, a mature neuron. And it will tell you what genes are expressed in both cell types and what ones are not. That's an extremely powerful technique. With one set of experiments, it allows you to detect the expression of every single gene, or at least what of all the genes are expressed in that particular cell or tissue type, all in one fell swoop. And to at least within a certain range, it's quantitative. In other words, it allows you to determine how much, what the level of expression of each of those genes is. There's certain limits on it. They can tell you whether it's expressed a little, expressed a lot, what have you. So the power of this technique is quite astonishing. It can tell you what genes are being activated, what genes are being shut off, what genes are being ramped up, what genes are being ramped down between cells in two different stages of development. Big on cancer research now. Because what you do is you take normal cells and then cancer cells at the same time. Let's say we take normal breast ductal cells and then you take breast cancer cells. And you do microarrays and you say, what genes in the breast cancer cells are different? They're expressed differently. Do you see in breast cancer cells certain genes expressed that are not in the normal cells? Do you see gene expression ramped of one cell, of one gene or another, ramped up or ramped down? Do you see genes that are expressed in the normal cells but not in the cancer cells? Those kinds of questions give you a molecular fingerprint of a particular type of cancer cell. And what that means is once you start identifying what genes you're dealing with that are misexpressed or not expressed or expressed where they're not supposed to be, that gives the drug companies targets to say, hey, these genes are involved in this type of cancer, maybe we can do something about that. So that's where, in fact, that's where the future of cancer therapy is going to be lying. Identifying the specific genetic defects or regulation defects and then targeting those things. So that's where the future is coming. It'll still take a long time before all that stuff's in the doctor's office. Some of that kind of stuff is in the doctor's office here and now. Depends on the type you have. So at any rate, it's a powerful technique. We don't have the capability of doing that here yet, but it's a very high-powered technique. Okay, so that's one cool thing. That's a relatively recent development, but it's extremely powerful because it allows you to detect the expression of all kinds of different genes all at once. Now, one or two more things, also molecular techniques, they're very important. First of all, you have the things where what we sometimes call recombinant DNA, or gene splicing is a common term for it. Okay, basic idea behind that is you're going to take and cut out pieces of DNA and insert those pieces of DNA, possibly containing genes or things like that, into other organisms. Or you can modify a gene by basically cutting out a piece of DNA and putting in a piece of DNA with a different sequence, deliberately introducing a mutation into that DNA and seeing what happens. Or with other techniques, you can effectively delete a gene or trash it so badly that it can't possibly be expressed. There's all kinds of cool things we can do with this. And of course, there's a lot of drugs out in the market now that are human proteins that are being produced by bacteria. For instance, most of the insulin that's used nowadays is human insulin. It used to be the old kind of insulin you got. It was extracted from cattle. Well, the cattle insulin is a little different than human insulin, and some people would get nasty and dangerous allergic reactions to it, which meant if you got diabetes and you can't use any insulin, you're in trouble. But now it's all human, not all of it, but almost all of it is human insulin. company called Genentech many years ago. They took the gene, they cut out the gene for human insulin, stuck in that bacteria, and got the bacteria to express that protein product, produce the insulin, and then they purify it, and there you have it. Likewise, hemophiliacs are missing, have a missing or damaged gene for a particular clotting factor that's necessary for your blood to clot. Well, used to be the only way you could get that is to take blood, is to extract it from blood plasma pooled from thousands of individuals at blood banks and stuff. Problem is, when they purified, they realized that often they were in that clotting factor concentrate that they inject in the hemophiliacs. It also contained the HIV virus. So for a while in the 80s and early 90s, the majority of hemophiliacs in this country and Western Europe ended up getting AIDS from that. Well, solution. Take that clotting factor gene, get in bacteria, have the bacteria express that gene. Bacteria don't get infected with AIDS. So now almost all the clotting factor is produced in bacteria. It's a human protein, but it's been expressed in bacteria. So a lot of these kinds of drugs on the market, these biotech drugs on the market, are human proteins that are being expressed in different organisms courtesy of gene splicing. Okay, basic idea here. Common thing, you get a little bit of DNA. You have to make lots of copies of that DNA. The major way they do that now is a technique called PCR. I don't have enough time to go into that here. Quick and dirty is that PCR, polymerase chain reaction, uses a device called a thermocycler that heats the DNA up enough so the strands separate. As it cools down, the DNA polymerase copies each strand of DNA. Now you have two strands in place of one. And then you repeat that for 30 or 40 cycles. So you take a piece of DNA, you start with one single strand of DNA, and after 30 rounds, you've got yourself a billion strands of DNA. And that's widely used in forensics too. That's how they have done things like convicted people of murder based on dried saliva on a cigarette butt thrown at the crime scene. That's how using a 2-year-old dress stain on the dress of a particular White House intern, they impeached our former president. And all kinds of different things. So it's widely used in DNA forensics, but it's a very good research tool because now you have lots of DNA and you can do all kinds of things with it. You can sequence it. You can cut out sections of it using what's called restriction enzymes. Restriction enzymes are enzymes derived from bacteria that recognize and cut only at specific sequences of DNA and nowhere else. Usually you're talking about sequences of DNA that are 5 to 10 bases long. It will not cut anywhere else. Change a single base in that sequence and the enzyme won't recognize it and won't cut there. That's why they're called restrictions. They're restricted to where they cut. They cut one place at one sequence and nowhere else. Well, that allows you to cut out pieces of DNA if you choose the right restriction enzymes. You could say, here's the insulin gene. And we cut it out with restriction enzymes. So now we got a sequence containing the whole insulin gene and hopefully some of the control regions. Okay, now we can take bacterial DNA, especially what they call plasmids, these little rings of DNA. Okay. And cut that with restriction enzymes. And then insert this new sequence in. And there's other enzymes that seal the ring of DNA up. And now what you've just done is you've inserted a human insulin gene into a bacteria. You grow the bacteria up. If you're lucky, the bacteria will actually produce the insulin protein. Now you purify it, and you make a huge amount of money in the process. So, and of course, we're not limited to doing that with bacteria. You can take genes from one organism and splice them into any other organism. For instance, somebody did a little experiment. They wanted to see whether this eyeless gene that is responsible in fruit flies for making eyes, they asked the question, they found that gene was only expressed in the area that was going to become the future eyes. So they asked themselves a question, what if we were to express that gene somewhere else in the fruit fly? So with a little bit of recombinant DNA, they managed to get a copy of that gene that they could activate very easily, and they put the eyeless gene that they could turn on or off at will in cells that were going to become the knees of the fruit fly. And then they activate the gene. And lo and behold, those fruit flies started growing eyes on their knees. And they looked like normal insect compound eyes. There were even evidence of innervation. Nobody could say whether they could see out their knees or not. But the structures were there and they were appropriate. So these kinds of things are pretty powerful. You could also, for instance, want to see, okay, well, what happens with a part of insulin? We kind of like cut this part of insulin out, of the insulin gene out. Would insulin still work properly? Or suppose we altered the sequence here. What does effect be? So people can do that, deliberately introducing mutations in a particular gene at a particular place, and then whatever mutation you want. Now that's technically difficult, but it's often used. And it can tell you the function of a certain part of a thing. So they do all kinds of things. Or what would happen if we deliberately put several copies of a gene in a cell or an embryo and have them all expressed, so overproducing that particular protein? What happens? Does it affect development? Sometimes it does. So using recombinant DNA is a powerful research tool. Of course, once again, it often ends up in the clinical labs as well. So one last thing I want to mention is also a relatively new technique. In fact, Dr. Murdoch does this quite a bit. There's a variant of PCR called real-time or RT-PCR. It's an interesting kind of technique. What it allows you to do is basically quantitate very precisely a particular messenger RNA, how much is being produced. What you have to do first is make a probe. And you have to label it, or have somebody do it for you. Okay. Now, so you've got this probe that recognizes specific messenger RNA. Okay, now what you do, you take your sample, cell, tissue, whatever, extract the total messenger RNAs, you make cDNA copies of all that, you amplify it using PCR, and while you're doing it, you add the probe at the early stages. Okay, and what it allows you to do is it tells you how much of a specific messenger RNA was present in your original sample. As you're copying the DNA, you'll get a curve, and you measure like the fluorescence. Most of these probes are labeled with fluorescent dyes. They glow in the dark. Okay, so what you see is as you're copying that, you get a curve kind of like so. Okay. And finally, you get, more and more. After you get to a certain amount of amplification, it's not going to glow much brighter. You've already kind of saturated the detectors and stuff like that. So you get a curve that's sort of like so. Okay. With some mathematics and stuff, you can look at all the aspects of this curve and give a very highly accurate number for how much of that messenger RNA was present in the original sample. So this is another, in fact, this is probably the most common way now of looking at the expression level of a particular gene. Now, I can't do that for everything all at once. You have to do it, you're looking at specifically. So you want to say, when is the eyeless gene first expressed in the area where the eyes will form in fruit flies? You can use that RT-PCR and look at that at all kinds of stages of development and say, oh, well, the eyeless gene is expressed immediately before the eyes start to form, or it's expressed long before the eyes start to form, whatever the case is. So that's a powerful technique. So like I said, it is done here. Dr. Murdoch does that for some of his research. So this is a quick and dirty overview of a wide battery of powerful new techniques used to investigate development of cells and organisms. And that's where we'll leave also. See you folks on Tuesday. Don't forget, very likely we're going to have, in fact, it's probably almost certainly, we're going to have a quiz based on our lab class on Tuesday. Okay, folks, so have a good weekend.